Good evening, advanced biology students. This is Mr. Jones. It is about 10:30 at night, and I'm going to try and get this lecture done before I finish my cup of sleepy time tea here. So anyway, we're doing our enzyme lecture, and we know that enzymes are a biological catalyst, and a catalyst is something that speeds up a chemical reaction. And we know that enzymes are part of the macromolecule group called proteins. So when we talk about metabolic reactions, we're looking at reactants and products. And when the reactants have more energy than the products, energy tends to be released from that reaction. That would be an exergonic reaction. And when the products require more energy than the reactants have, you need a reaction that is going to absorb energy. That is endergonic. So overall, most reactions do not occur spontaneously within cells, and therefore would need some type of biological agent, such as an enzyme, to speed them up. So we know that some processes in the cell use ATP, which is the energy currency of cells, abbreviated ATP, which from its name, adenosine triphosphate. So how does it function? Well, it does chemical work, which means it supplies energy needed to make macromolecules that make up the cell and the organism. This we could see like when things are being synthesized in the cell. It does transport work, supplies energy needed to pump substance across the membrane. We learned that last chapter, active transport. It also does mechanical work. It supplies energy needed to make muscles contract and other cellular parts their ability to move, like flagellum. Flagellum would be, you can see that whip-like structure on some prokaryotic cells, some animal cells like sperm. When that flagella is moving, it requires an input of energy. We discussed ATP when we did nucleic acids. We said that ATP is the high energy rich molecule of our cells. That bond, that terminal bond there between phosphate number 2 and phosphate number 3, when broken, releases energy, and that would be an endergonic reaction because energy is being released, and you form the molecule ADP. ADP is adenosine diphosphate. ADP and ATP are like rechargeable batteries, in that once one is consumed, it can be reformed again. So metabolic pathways and enzymes. An enzyme is a protein molecule that functions as an organic catalyst to speed up reactions. So if you look at an enzyme, you would picture that protein all folded up into its primary, secondary, and then the functional levels, tertiary and quaternary protein level. That's what makes up an enzyme. And those foldings create this little site on it called the active site. And the active site is where the substrate's going to bind. The substrate to an enzyme is the reactant in a chemical equation. So reactants in the enzymatic reaction, this is what an enzyme attaches to. So basically what enzymes do is they lower the energy of activation. There are two types of energy that we're going to look at here. There's the free energy, and that's the energy that is available to do work. And then you have activation energy. And activation energy, or also termed the energy of activation, that is the energy that's required to get a reaction going. So what enzymes do is enzymes will lower the energy of activation. So if you look here we see that enzymes are made up of proteins. They speed up chemical reactions inside the cytoplasm. They are needed only in small amounts. So they are only needed in small amounts. They remain unchanged after each reaction, can be therefore reused. So they are not consumed in a chemical reaction. They can be recycled and reused and reused and reused. And more importantly, each enzyme is specific to its substrate. So each enzyme carries out its own individual part of a chemical reaction within an entire metabolic pathway. So if you look there, here we have the cycle of enzymes. We have one, the enzyme and substrate are available. Then the enzyme here, so we have our enzyme called sucrase, and then we have the active site. And if you look at the substrate, the substrate right here, that's sucrose. Sucrose is going to come in. Here you have the enzyme substrate complex right there. And if you look, it fits in perfectly. So that substrate fits into that active site perfectly. It's much like the lock key model. So the substrate will bind to the enzyme. Here we have water coming in, and the substrate is converted into the products of glucose and fructose. And those products will be released, and then you'd have the enzyme sucrase ready to be used again. For those of you that recall from biochemistry, this would be a hydrolysis reaction. Water is split, breaking the bond there, where that disaccharide is broken up into its monosaccharide subunits. There are two methods that we look at when thinking about enzymes. We have the induced fit model. And in the induced fit model, the substrate and enzymes fit together like a lock and a key. You have the degradation versus the synthesis, breaking down versus building. So degradation, you have the substrate binding to the enzyme, and then that enzyme substrate complex, the products are released, and the enzyme then moves forward and is recycled. So this would be the degradation. So you're breaking down that reactant into its individual products. In a synthesis reaction, such as like dehydration synthesis, you have the substrates binding to the active site of the enzyme, and then you have the process occurring where you form that bonds, and then the product will release. So you're making something there. So factors that affect enzymes are enzyme speed, would be substrate concentration, temperature and pH, and enzyme concentration. Enzymes can be denatured, therefore they change shape so much that they are no longer effective. A high temperature or a pH can cause denaturation. So you'll end up losing that induced fit, that whole lock key model scenario, where each substrate is specific to its enzyme, much like each key is specific to a lock, and that induced fit where that key just fits perfectly right into that lock. So if you denature the protein or the enzyme, what happens is you no longer have that induced fit where it kind of just fits in there perfectly. The enzyme is unraveled and therefore is no longer rendered effective. And that's it for today.